As we all know, drugs that help you lose weight are very, very hot topic at the moment. Evidence shows that when you stop taking those drugs, your body weight simply increases back to what it was before. One of the biggest challenges at the moment is obesity and diabetes. And those are probably not because we eat too much when we are hungry, but because we eat too much when we are not hungry. So I'm Korneel Hens. I'm a senior research fellow here in Oxford Brookes. My job is to do research and also to do a bit of teaching, mostly about the research that I do. And my main research interest is to look at our genome and to see all the genes that are involved in regulating feeding behavior and how the diet influences which genes are activated or inactivated. A gene is really a bit comparable to the plan you get when you build something in Lego. But the way you assemble things and which bricks you need at what point, those are all encoded in our genome. So the genes tell us what our bricks are, but also when you should use them and where you should use them. If we have a better understanding of how these changes in behavior are regulated, because if we can then intervene with that, that might help people keeping a healthy body weight. The day-to-day work that we do is we would select a fly line that we have created, where we have either artificially activated the gene or artificially inactivated the gene. And then we can look at the results of that. This can be just simply looking, does it eat more or does it eat less? But maybe more interesting is, does it change its preference? So what we would, for example, do is we put different food choices in front of the fly, each colored with a different dye. And that way we can see if it prefers sugary food, if it prefers fatty food, if it prefers food with a lot of proteins. And in that way we hope to determine which genes code for certain preferences for certain macronutrients. And that can give us an idea of which genes are involved in what illnesses or diseases in humans. So the reason we use fruit flies is because you might not think it, but they have a lot in common with us. 60 to 70% of all the genes that can cause diseases or illnesses in humans, we can also find them in fruit flies. I worked with an art therapist, which was a really, really interesting experience for me. The aspect of my work that we explored was how it is as a PI or a principal investigator to run a lab, to be responsible for junior researchers, to make sure they find their ways. So they all have their hopes and dreams. They all come in with a certain idea of what science is. It's a lot of managing expectations, it's a lot of managing disappointment, but also a lot of celebrating successes that we have. What we did was we tried to depict that aspect of my work as a maze where you can go down certain pathways. Some of them will be a dead end, some of them will lead to success, but via certain disappointments or disagreements. It really helped me to reflect a bit more on the type of group leader that I want to be and how I want to interact with my people. The most important thing is that scientists are only human. We all have our hopes and our dreams and our disappointments, and it takes a lot of hard work to achieve what we do. For me, the science and the discovery makes that all worthwhile. The successes are far outweigh the disappointments. Look around you and ask, why are things happening the way they are? The question why should always be forefront in your mind. If you go out for a walk and you see the insects or the birds, or why do they do what they do? How does that work? And that's the correct point of view.